Good morning. Apologies for the slight delay. It's my honor this morning to introduce our first speaker, Dr. Suzanne LeBlang, who is the Director of Clinical Relationships and Co-Chief Medical Officer at the Focused Ultrasound Foundation. She's a board-certified neuroradiologist who earned her Bachelor of Science and her MD from the University of Miami's honors program in medical education. She's one of the leading experts in the clinical application of focused ultrasound, and she's going to be presenting to us remotely. So you can clap if you want, but she probably won't hear us.
Great. Good morning, everybody. So thanks so much. I really appreciate the opportunity to share what's happening with focused ultrasound and Alzheimer's disease with you all. And our mantra at the foundation is, it's going to be the most powerful sound you will never hear. So as you know, Alzheimer's disease is the most common neurodegenerative disorder. And although the pathogenesis is unclear, the amyloid cascade hypotheses remain the prevailing theory, and the deposition of beta amyloid is likely a pivotal initiating event. So to date, as you know, there are no great treatment options. To treat the symptoms, we have some cholinesterase inhibitors and glutamate regulators. For disease-modifying therapy, there are anti-amyloid antibodies, which have been shown to reduce beta amyloid and slow cognitive decline. However, they are not great. Many patients are ineligible for treatment. The treatment itself is associated with side effects, and the antibodies do not cross the blood-brain barrier efficiently. So the question is there another approach that could act synergistically with existing treatments to make it better? So for the case study, I'd like to share this picture with you, because as a neuroradiologist, I love to share pictures. This comes from a recent journal article in the New England Journal of Medicine. And if you can see on the right here, this is an amyloid scan, and the red is the amyloid deposited in the brain, mostly in the frontal lobes. Okay. So the trick is, can we decrease the amount of amyloid and improve cognitive symptoms in this patient, right? So just think, if you give an anti-amyloid antibody to this patient, in theory, it should kind of go everywhere in the brain, but we know it doesn't cross the blood-brain barrier efficiently. So now let's think about if we have a procedure such as focused ultrasound, where we could deliver this technology, let's say, to the left side of the brain here, and this area in green is where they're going to target the area. I want you to think about the scan I'm going to show you later on in this presentation, and what's going to happen to the amyloid in the left frontal lobe of this patient compared to the right side. And remember, the anti-amyloid antibody is supposed to go everywhere, but just the focused ultrasound is going to go to the left side. So think about what that picture is going to look like in your mind. So the objective for today is I want you to help you understand the different applications of various forms of low-intensity focused ultrasound in Alzheimer's disease research. This is going to involve blood-brain barrier opening alone, blood-brain barrier opening for drug delivery, and neuromodulation. And I also like to point out that there are significant barriers to our clinical research, including the comparison of different focused ultrasound machines, confirmation of target engagement, and standardization of both clinical and radiographic outcome measurements. So what is focused ultrasound? Well, it's really cool. It's a way to do incisionless changes inside the brain. So unlike diagnostic ultrasound, where you take a probe and the ultrasound beam fans out to create an image, we're doing the opposite with focused ultrasound. We're taking a beam of focused ultrasound, and instead of it fanning out to create an image, we're making the beams converge at a focal spot. And these machines are so advanced that they can go right through the skull and form a target area in the brain where we can have a bunch of bioeffects. And remember, there's no incisions. It's ultrasound. There's no radiation. There's no implanted wires. There's no risk of infection or bleeding, and the patients are completely awake during this procedure. So it's kind of like when you were young and you used to go out in the sun and you took a magnifying glass and you made all the rays converge at a spot on the leaf, and you can make the leaf catch on fire. So it's the same thing. Instead of the sun's rays, we're using the ultrasound beam. The other difference, which I'm going to tell you about in a second, is that we don't have to just make this beam strong enough to create heat for it to catch on fire or cause thermal ablation. We can use it in a lower energy and have different bioeffects. So I thought this is a nice graphic that we use at the foundation to help illustrate this. So there are thermal mechanisms of action of focused ultrasound and mechanical mechanisms of action of focused ultrasound. And with each of these, we can use increasing power. So thermal ablation is the most well-studied mechanism of action of focused ultrasound, and it's approved around the world for over 32 different clinical indications, over 9 in the United States. But in the brain, it's actually FDA approved to treat essential tremor Parkinson's disease, where we can create a small lesion in, let's say, the VIM nucleus of the thalamus to control the movement disorders. Okay. But what we're going to talk about today is this lower left-hand corner. We're going to use mechanical mechanism of action of low intensity focused ultrasound for blood-brain barrier opening and neuromodulation. Also note that there are some immunomodulatory components from all of these things that are also included. So this is a very basic video, but I wanted to show it to you. So here are the endothelial cells lining the blood-brain barrier. You can see the tight junctions over here. The purple is going to be some kind of drug. It could be a chemotherapy, antibody, gene vector. And the red circles here are actually microbubbles. They're FDA-approved microbubbles that you inject intravenously. And what's going to happen is that in the presence of a focused ultrasound beam, these microbubbles are going to oscillate. They're going to get bigger and smaller and bigger and smaller in a very controlled fashion called stable cavitation. And they're going to push on these tight junctions and open them so that the drugs can go in a targeted fashion only where the focused ultrasound beam is opening these tight junctions. So you can see the bubbles oscillating, the tight junctions open, and the drugs go through only in the area of the focused ultrasound beam. We're very, very lucky that we're going to hear from Dr. Scott Burks after me, who's going to really describe the mechanisms of action of how this blood-brain barrier opens, including the opening of the tight junctions, the increase in transcytotic vesicles, and a decrease in the efflux pumps, all helping to deliver medicine. And he's also going to go over the bioeffects with a sterile inflammatory response. There's microglial activation, astrocyte reactivity, release of cytokines and growth factors, and no neuronal damage from the blood-brain barrier opening. So I really look forward to hearing his presentation after. So in addition to just blood-brain barrier opening alone, which creates bioeffects, we can also deliver the drugs. Again, these can be antibodies, other therapeutic agents, and in the beam path will help deliver them. Now, if you want to know the history of delivering drugs with focused ultrasound, I refer you to this review article that was published last year with anti-cancer drug delivery by focused ultrasound for glioma therapy. And there are numerous clinical trials, hundreds, probably almost 1,000 treatments worldwide now, where we've opened the blood-brain barrier to deliver drugs to gliomas. We found that we can open the blood-brain barrier safely, and it's reversible. It closes within 24 to 48 hours. And there's been different therapeutics for the glioma space, such as carboplatin, TMZ, Abraxane, liposomal doxorubicin, and importantly, Avastin, which is an antibody to these brain tumors. And the reason I mention the Avastin is because it's about 150 kilodaltons. And if you look over here, antibodies that are about 150 kilodaltons normally do not pass through the blood-brain barrier, right? So that's the issue now. We're giving these anti-amyloid antibodies, but are they really getting in the brain in sufficient quantities to do significant, to provide significant help to the patients? So let's see how we can do this with focused ultrasound. There are a variety of machines in this space. So there are machines that are performed inside an MRI machine. So the Exablate machine is made by Insightec. It has over 1,000 elements around a cap around your head, and you do it inside the MRI machine, which provides you with great anatomical guidance by looking at the images and target engagement right away, because we use gadolinium as a surrogate for blood-brain barrier opening. So where you want to open the blood-brain barrier, you can inject gadolinium and confirm that you are actively doing that in that area. There are other machines, such as the Navifus machine and a device at Columbia University called the Delsona, which uses neuronavigation. So they take a previously performed MRI scan, put it in a computer system, then the patient can sit outside an MRI scan. They can sit in your clinic, they can sit in an office, and through optical guidance, they can place the focused ultrasound machine over the patient's head and direct it into the target that you see on the previously performed MRI scan. There was also a company called Carthera that made a SonoCloud machine. And what they actually did was they implant the focused ultrasound transducers in the skull. They're usually doing this for brain tumors because the postoperative bed is obviously static and not moving. So they put the transducers right over the tumor in the post-op bed. They did do a study for Alzheimer's disease, which I'll tell you again about later. So we went over blood-brain barrier opening alone as a mechanism of action, blood-brain barrier opening for drug delivery as a mechanism of action, and there's also neuromodulation. So there have been numerous papers trying to investigate what the exact mechanism of action is for focused ultrasound and neuromodulation. It's not completely understood, but it's likely related to changes in the calcium influx along the cell membranes due to this intramembrane cavitation. Most often, this is done without microbubbles. It's just a focused ultrasound beam alone at low power. It can also activate NMDA receptors via mechanosensitivities and increase neurotrophic factors in the brain. The machines in this space are also varied, with some of them used inside an MRI machine, like the BrainSonics machine and the InsightTech machine. Others are neuronavigated, as I explained to you before, such as the Neurosona machine, one in Australia from Ultrathera, the Storz machine, and some of them have no guidance at all, where they're just going through the temporal lobes and hitting some important parts of the brain with Alzheimer's disease, such as sound wave innovation. So again, there's a lot of interest in the space by companies. So let me tell you how this story began. So back in 2014, 2015, the Focused Ultrasound Foundation noted that 2 different labs in 2 different areas of the world published very similar results, and each one didn't know what the other one was doing at all. They showed that doing focused ultrasound to mice models decreased amyloid and improved cognition. So we said, something's going on here. We really need to convene the community. We held a workshop later that year where we invited preclinical researchers, clinicians, regulatory experts, manufacturers, and nonprofits. And we sat in that room and we said, what additional preclinical studies need to be performed to move this field forward? And what does the first in human clinical trial look like? Are we ready yet? So after we accumulated some additional preclinical evidence, which included from blood-brain barrier opening alone, that the studies showed that you could reduce beta amyloid with enhanced microglial and astrocytic phagocytosis, IgG and IgM co-localized with the plaques. We were able to reduce tau. We were able to show that you could elicit trophic factors to promote hippocampal neurogenesis and activation of intraneuronal signaling. There was results in cognitive improvement in these preclinical animal models, and it was safe with no bleeds. So great, blood-brain barrier opening alone showed a lot of promise. At the same time, other researchers researched blood-brain barrier opening with therapeutic drug delivery, and they showed the increased delivery of Aduhelm 5 times as much as without the focused ultrasound, and it resulted in improved spatial memory and reduced amyloid. Others were able to show an increase of Aduhelm delivery of almost 8 times with less cognitive decline and also reducing amyloid. They showed increased delivery of anti-tau antibodies, increased delivery of IVIG, which also reduced pro-inflammatory cytokine TNF-alpha and showed inhibition of neurogenesis and promoted hippocampal neurogenesis much more than the IVIG alone or the focused ultrasound alone. So you can see what I'm getting at. We can improve drug delivery and improve outcomes. And again, it was shown to be very safe with no bleeds. The preclinical models that I just talked to you about, these were 2 of the studies. This one showed the effects of aducanumab and scanning ultrasound on amyloid plaques. And what you could see here on the left side is, here's the sham model with lots of amyloid plaques. With Aduhelm alone, you had some decrease in the plaques. With scanning ultrasound alone, you had some decrease in plaque. But look at the difference when you did the scanning ultrasound and the Aduhelm together, a much more dramatic decrease in plaque. So we showed the decrease in plaque. We showed almost 5 times higher levels of Aduhelm in the tissue compared to without the focused ultrasound, and improved spatial memory. And here you can actually nicely see that the Aduhelm shown here in the right-hand corner nicely overlapped with the microglia in the location of the plaque, which is exactly what you want. So this preclinical study was very hopeful that we could deliver drugs with focused ultrasound. And it was recapitulated in this experiment as well, where they interestingly used a lower dose of Aduhelm because, of course, we want to decrease the side effects of getting too much Aduhelm in the brain. So they used a lower dose and still got over 8 times as much drug delivery into the brain as without the focused ultrasound. And it resulted in restored cognitive impairment and significantly reduced plaque. So After all that stuff was done, they were ready for the first in-human clinical trial, and this was the result of 5 patients, and it was completed in 2018. You can see here each column showed a different patient. Row A is the amyloid scans before the focused ultrasound. So you could see the asymmetric amyloid deposited in this right frontal lobe. After the focused ultrasound procedure, while the patient was still in the scanner, we gave gadolinium and confirmed that we opened the blood-brain barrier as evidenced by gadolinium enhancement, exactly where we opened the blood-brain barrier in the region of the amyloid plaques. And then approximately 1 week after, they did the amyloid scans alone, and you could see there was a slight decrease in the amount of amyloid in the targeted regions. So importantly, in this first in-human clinical trial, we showed we could open the blood-brain barrier safely in Alzheimer's disease patients. There were no bleeds, and there were slightly encouraging results that we were able to decrease amyloid in these regions. Over time, additional centers at additional institutions succeeded in opening the blood-brain barrier in other areas that are implicated in Alzheimer's disease. So they started targeting the hippocampus, where you could see the contrast enhancement in the region of the blood-brain barrier opening. They were also able to show amyloid plaque reduction. Additional studies all around the world were showing that they can open up larger areas of the brain and involving multiple sessions, because this is probably not a one and done procedure, right? So in this study, this is before focused ultrasound in the top row, after focused ultrasound in the bottom row. And again, you could see the areas in red that had higher SUVs with amyloid decreased after the procedure. And again, they showed it was done safely. So again, very encouraging results. And in this chart, I wanted to kind of summarize the clinical landscape for you. And there's a lot of important data in this chart. So let's start with the left-hand column. They started opening a very small volume of tissue. And as you can see here, as the studies progressed, they were opening larger volumes. It started with just a few sessions, and then various studies repeated the numbers of focused ultrasound opening sessions over varying time periods. In terms of the stage of Alzheimer's disease that they were treating, you could see the variation of mild to moderate AD, moderate to severe AD, with varying levels of MMSE numbers as inclusion criteria. So there's been wide variability in the stage of disease that's being treated in clinical trials right now. In terms of the target locations, some are targeting the frontal lobe, some at the hippocampus, some along different parts of the default mode network. So again, variability here. You can see the different machines that I described to you before are in clinical trials. And importantly, what they're noting as imaging outcomes and clinical outcomes is also quite different. So let's do the clinical outcomes first. Some are just looking at expected changes, various clinical AD scales. In this particular study, they showed a decrease in the caregiver neuropsychiatric inventory scale. There were no changes in MMSE scores that were lowered, but this study over here showed no cognitive decline beyond the expected 1-year MMSE changes and ADAS-Cog. So you can see there's great variability here, but some suggestion of improvement with the CGA and PI scores in 2 of these studies. Now, in terms of the imaging outcomes, some of them demonstrated a lower A-beta load increase in the treated area. Some of them noted a 14% decrease in centiloid units and a 5% decrease in SUVR. Some had a 32% average reduction in the targeted area. And so you can see there's variability in reporting here. It's not standardized. Importantly, in terms of adverse events, in this study, there was a transient amount of edema on MRI in 1 patient that was clinically insignificant, and 1 patient in this study that had some edema and T2 star changes that was asymptomatic and resolved. So overall, in all of these clinical patients, it has been shown to be safe. So that was blood-brain barrier opening alone, except for this study right here, which is bolded, where they started with aducanumab, and when that was taken off the market, they changed to lecanemab. So this is the one with blood-brain barrier opening for drug delivery, the only one so far. Okay. So this is the study that was published from that report in the New England Journal of Medicine. And this is a graph from participant number 3, which shows that the reduction in the amyloid changes was greater in the regions targeted with focused ultrasound than in the contralateral region that was not exposed to focused ultrasound. And here's that picture I showed you at the beginning. Here's where they targeted the focused ultrasound. You can see we had contrast enhancement there, indicating successful blood-brain barrier opening, which closed after 24 hours. But look at the scan. This is really incredible. Look at the area that was targeted with focused ultrasound compared to the side that was not. That's a dramatic asymmetric decrease. And you can assume it's due to the focused ultrasound because the patient received the aducanumab, and in theory, it was going everywhere in the brain equally, although to a very little degree, because we know it doesn't cross the blood-brain barrier opening alone, blood-brain barrier alone. So this is really a dramatic picture of what the addition of focused ultrasound can do. Now, in this review, it really nicely went over the fact that you could do the blood-brain barrier opening alone with the microbubbles. You could do it for drug delivery. But what about doing focused ultrasound alone as a neuromodulatory component? So it turns out that doing scanning ultrasound alone can restore some cognitive function in mice and doesn't necessarily need the removal of amyloid. So there's likely multiple mechanisms of action happening about how focused ultrasound affects patients with Alzheimer's disease. And this was that study. It showed focused ultrasound alone improved cognition and memory without removing amyloid. They tested 2 different frequencies of the transducer and showed that the 1 megahertz transducer was better than the 286 kilohertz, possibly related to increased in acoustic radiation forces. Remember, we talked about those neuromodulatory mechanisms of action and how it would probably change the calcium influx. And it also resulted in proteomic changes at the synapse level. It improved spatial memory in the APA avoidance test. On MRI scan, they showed increased neuronal connectivity and changes in microstructure on the diffusion MRI and showed it was safe. Again, here you see the high frequency group had fewer shocks on day 5 and had a better learning index compared to the lower frequency transducer. So again, really interesting that you don't necessarily need amyloid reduction for cognitive improvement. This was a recent review article that nicely outlined the preclinical focused ultrasound effects in animal models versus the effects that we've noted to date in human clinical trials for blood-brain barrier opening with a therapeutic, and the LIFU neuromodulation. And remember, the mechanism of action is probably multifactorial: mechanical effects on membrane and ion channels, increased blood flow to the area, increased neurotrophic factors. Again, you can improve cognition with or without the decrease in amyloid. And again, the FUS advantages are that there's no incisions. It has a very high spatial precision of millimeters if you want to target very specific areas. And we can target superficial and deep regions, which are benefits compared to other non-invasive procedures. So this is the clinical landscape for our neuromodulation clinical studies. Again, it goes from doing 1 session to multiple sessions. The MMSE scores are here, are quite varied from mild AD patients with MMSEs over 10 to those that are just less than 20. So, there is variability here. They're targeting different areas of the brain. Again, I showed you the different machines before. With respect to imaging outcomes, they're showing improved fMRI results with improved connectivity. There's also been shown to have clinical outcome improvements in the ADAS-Cog scales, CRAD scales, and actually importantly up here, the Neurosona reports mild improvement in the MMSE score, executive and global cognitive function improvements, and significant improved memory with recognition and recall. So again, this is really exciting. What is slightly different about this neuromodulation study compared to the other ones is that they also used microbubbles with the lower energy levels of focused ultrasound neuromodulation, but they did not do it intentionally to cause blood-brain barrier disruption. In fact, they didn't want it. And they proved with gadolinium that even though they added the microbubbles, they did not get blood-brain barrier opening. So that was not a mechanism of action. but maybe there are other mechanisms of the microbubble doing something to the endothelium that's also helping as well without doing the blood-brain barrier opening. So this is going to be very interesting to follow. So let me show you some results from two of these clinical studies. This is the sound wave innovation study. It uses no image guidance. And it showed that the worsening of the ADAS-J-COG scores was suppressed in the low-intensity focused ultrasound group compared with the placebo group, meaning that the LIPUS group did not progress as quickly as the placebo group. So that's encouraging results. And with the Neurosona machine, using the neuronavigation aspects of MRI scan, their hypothesis was that impairment in verbal learning and memory is the earliest finding in AD, and it's associated with hippocampal dysfunction. So they targeted the hippocampus, they showed increased blood flow in this region, and an improved verbal memory and recall, and that also correlated with the amount of glucose metabolism. So the more glucose metabolism in that area, the better the patients did. So they actually just finished their second pilot study, and the results are going to be published hopefully soon. So back to our case report. I hope that you now see that focused ultrasound can really add tremendous benefit in addition to giving the anti-amyloid antibodies by opening the blood-brain barrier, increasing drug delivery, which resulted in decreased amyloid in that region compared to the untreated side. I hope you understand now that there are different applications of forms of focused ultrasound, such as the BBB opening alone, BBB opening for drug delivery, and neuromodulation. And hopefully I've identified the barriers that are in our clinical research. The focused ultrasound machines use different frequencies. Some are 1 megahertz, some are 256 kilohertz. So how do you compare the results if they're using different frequencies, different volumes of targeted tissue, different areas of targeted tissue, different outcome measurements on clinical scales, and different radiographic analysis of even the amyloid scans? So we really need to help the community standardize some of these reporting outcome measurements and parameters so that we can start to compare the results across the different machines and across different patient groups. For the future, I want you to start thinking about a few things. Number one, we can certainly improve the focused ultrasound technology. How about creating wearables for patients to ease repeat treatments? How about devising specialized transducer caps based on the patient's individual skull to optimize the transmission of the focused ultrasound beam into the target area? How about using ultrasound guidance for targeting and feedback compared to MRI scan? And Dr. Konafagu's lab is really onto some really interesting results of having 2D and 3D cavitation mapping to look real time at targeting. How about using AI for patient selection and predictive modeling? Who are the best patients? Using liquid biopsy and radiographic data, right? Which patients are going to best benefit from BBB opening alone, BBB opening with drug delivery or neuromodulation, or a combination of them? How about when to treat? What stage of disease, right? Are we going to really be able to change disease in the late stages by removing amyloid or doing neuromodulation? The data is still not out there yet. However, there was a report out of Dr. Konafagu's lab at Columbia that showed cognitive and pathological protection for asymptomatic carriers of AD mutations in preclinical models. So wouldn't that be so exciting if we could use liquid biopsy or radiographic data or genetic data to see who have very early stage disease or who are carriers and start to do some of these things to protect downstream changes, right? That would be super interesting. And of course, the Holy Grail may be doing advanced research with FUS-BBB opening and gene therapy. So maybe we can be one and done in treating these patients. So I want to thank you for your time and your interest in this subject. If you have any questions, please contact me at sleblang@fusfoundation.org. You can go onto our website. We have a newsletter that comes out and is full of this really interesting information. So thank you for listening.
Thank you, Dr. LeBlang. Dr. Naff has a question.
Yeah, I really enjoyed your presentation. So it's very impressive as to how the ultrasound can actually dissolve these amyloid plaques, which we usually think are very rigid in the brain. It suggests that maybe they are much more friable than we originally thought. But how long would it take for it to reaccumulate then? If you get rid of it, and do they come back quickly? Does it take a long time to accumulate? What happens to them?
Yeah, that's very interesting. And I think that's why the clinicians are studying how often they need to repeat these sessions. Is it once a week? Is it once a month? Is it biweekly? I'd have to go back to the preclinical models to see if they evaluated the reaccumulation and it's very expensive to do the amyloid scans, so they really are hesitant to repeat them to see how often it comes back before they have to repeat it again. But I'm happy to look back at the data and see if that was done. But I know they're doing the increasing sessions because they know it's not just one and done. They will reaccumulate. It'll also be really interesting to see if there's faster reaccumulation with the neuromodulation versus the BBB opening. So I think those are lots of really interesting questions.
All right, thanks.
Dr. Hallett?
Question about, do you have any effect on tau as opposed to amyloid?
Yeah. Believe me, I'm a neuroradiologist. I could talk to you for 8 hours about this subject or more, and I just decided to concentrate on the amyloid. But there are preclinical studies out there that have been published. You can look them up that show that the focused ultrasound decreases tau as well. And there is a first-in-human clinical study now where they're doing amyloid and tau imaging after focused ultrasound BBB opening to see whether the tau gets removed equally, as quickly, or better than the amyloid, and what that effect is on cognition as well. So we have our first in-human clinical trial starting with that right now. Good question.
Thank you again, Dr. LeBlang. Great presentation. Now I'm going to introduce our second speaker today. Dr. Scott Burks is a senior associate scientist in the Frank Laboratory here at the NIH Clinical Center's Department of Radiology and Imaging Sciences. He earned his Bachelor's of Science at the University of New Mexico, then his PhD at the University of Maryland School of Medicine. He joined the Frank Lab as a postdoctoral fellow and hasn't left NIH since, except for lectures. He became a staff scientist and is now senior associate scientist. He's published broadly on applications of pulse-focused ultrasound in preclinical models and also has a patent on targeted delivery of imaging probes for in vivo cellular imaging. Please welcome Dr. Burks.
Thank you very much, Christy, and thank you again for inviting me. I'm really happy to be here and share some exciting findings with everybody. I would also like to thank Suzanne for a wonderful talk. I also have to note that she was recruited at the last minute, so I'm extra appreciative that she was willing to join us today and step up and show what you see really is a lot of potential for this technique. What our group is interested in is looking at the intersection of biology and physiology and pathology by extension with the therapeutic ultrasound modalities. So today I'm going to discuss our work in blood-brain barrier opening. And I'll start by giving a background of blood-brain barrier opening with focused ultrasound. I'll try not to repeat and be redundant with some of the background that Suzanne gave. We have done a lot of studying on the neuroimmunological changes, both cellular and molecular, as well as in the acute and long term. What I hope to show you today is that we see that these bioeffects from the focused ultrasound blood-brain barrier opening contribute to a lot of the improved outcomes that we see in pathology. And lastly, there is a nascent line of research that I think is very interesting in our lab right now, demonstrating the intersection of endothelial cell physiology and blood-brain barrier opening. Okay, so therapeutically, we have numerous strategies to bypass the blood-brain barrier if we want to deliver drugs. Obviously, things like direct injection or intravascular infusion of pro-inflammatory agents, intravascular administration of hyperosmotic agents like mannitol. There's a whole host of highly experimental techniques, as well as using focused ultrasound to open the blood-brain barrier. Okay. Again, just to sort of go over this at a high level, since Suzanne covered it so well, is that, again, it's these intravascular microbubbles. These are diagnostic ultrasound contrast agents, but we infuse them and then they undergo a process called cavitation when they are in the ultrasound field. And that disrupts tight junctions and allows passage of a number of different molecules. We can get small molecules through, a lot of different sized dextrans have been shown to go through, and then large molecules such as Evans blue, which comes through on albumin, or antibodies. And one of the things that I want to point out is that there is a large frequency dependence on the bioeffects that we get, and that focused ultrasound does occur over a range of frequencies. And so with things like 0.2 megahertz would be what we would see in like the human trials, but preclinical systems may go up to even megahertz or even higher. Okay, so when we got interested in the bioeffects of blood-brain barrier disruption, this is sort of what the landscape looked like from a preclinical standpoint. So here you can have you lay the rat down in a bed of water, you use MR imaging, and you can target precisely specific regions in the brain. We see that Evans blue leaks across, and electron microscopy shows that, in fact, that these leakage pathways occur through paracellular transport. So this is not transcytosis, but rather leakage through the tight junctions. There are some observed hemodynamic alterations. So there is in the immediate or during the FUS, you get vasoconstriction. There is some vasospasm, but that's associated with the blood-brain barrier being open. And there's also a lot of downregulation of tight junctional proteins, as well as activation of certain cell signaling pathways like GSK3 and AKT. Okay, so we published this study a few years ago where we looked at the molecular responses following blood-brain barrier disruption. So we did this in young female rats, and we used MR targeting to open many foci in the cortex, and then we would harvest the tissue at various time points, and then what we would do is multiplex ELISAs to examine changes in protein levels. And what we see here is that over time, there is a highly dynamic neuroimmunological microenvironment that's being changed. And we get lots of changes in things like heat shock protein, interleukins, interleukin 1, interferon gamma, and TNF alpha. And this is just a sort of a 10,000-foot view overview of how all the different cytokines, chemokines, and trophic factors change and evolve over time, with most of it resolving by 24 hours. Certainly within a couple of days, it's entirely gone. Okay. What this looks like is a sterile inflammatory response. Okay. This is a immunological response initiated following injury or necrosis, and is characterized by what we call damage-associated molecular patterns, or DAMPs. And DAMPs are analogous to PAMPs in pathological infection. And here is an example of it in ischemic stroke, where it starts with neuronal death, which then activates glial cells and ultimately causes blood-brain barrier damage. But what I want to point out is that a lot of these pathways actually work both directions, so that opening the blood-brain barrier, for example, can initiate many of these same molecular changes here. If we look at some transcriptomics, again, we see lots of changes associated with blood-brain barrier opening, that early on we get things like cyclooxygenase 2, we get NF-kappa B, and then persistently, again, in gene changes, we see interleukins 1s, lots of selectins, ICAM, so cell adhesion molecules, as well as TNF alpha. If we look for phosphorylated protein activity, we see, again, upregulations in phospho-AKT, phospho-GSK3, without activating MAP kinase pathways. Okay, so if we want to look histologically, what we do is we repeat this same treatment, and then we will harvest at different time points, so 1, 6, or 24 hours after HIFU. And one thing that we want to point out is that there is no evidence of microhemorrhage in these following this blood-brain barrier opening. We've done a lot of high-field MR on histology, so we're really not getting extravasation of red blood cells. But what we do see is extravasation of albumin, and that by 6 hours is where it really peaks, but then much of it is cleared within 24 hours in the treated side compared to the contralateral side. If we look at tunnel staining, so this is an enzymatic reaction that detects DNA damage or double-stranded DNA breaks. It's not necessarily equated with apoptosis or necrosis, but rather simply DNA damage. And we do see that there is some DNA damage associated with blood-brain barrier opening, but that does start to resolve within a day or so. We do get persistent upregulation of ICAM expression on vessels. And then we do get some increase in GFAP as well as IBA1 for reactive astrocytes. Okay. One of the things that we also want to note here is that there is some systemic effect to blood-brain barrier opening. If we look, we can label splenic monocytes using these fluorescent iron oxide nanoparticles. And what we can do is we can label these in vivo several days before we want to open the blood-brain barrier. So this only labels monocytes outside of the CNS. Then what we do is we disrupt the blood-brain barrier, and then we can come back a few days later and we can look by histology. And what we see is the presence of a lot of these fluorescent microspheres here. And in fact, that they are co-localized with CD68. And not to harp on this, but that was the point being is that these are external monocytes that are coming in. So this CD68 is not resident macrophage CD68, whereas in the contralateral side, we really don't see anything. So there is some communication with immune organs outside of the CNS. Okay. One of the things that we were really lucky to do is very fortunate to collaborate with Dorian McGavran and get some 2-photon microscopy of blood-brain barrier disruption here. And so what we can do here is we use 2-photon microscopy and we take images beforehand. We will take off the mouse and give the ultrasound with microbubbles and then put the mouse back on the microscope we can do immediately after, a few hours after, and a few days after. For what I would note here is that we use what's called LIPUS, which is low intensity pulsed ultrasound. And this is not focused ultrasound. So this is an unfocused beam, but the intensity is still on the order of magnitude that you would get from focused ultrasound. We just simply lose the ability to spatially focus in small regions. So for these sonications here, and you can see this Evans blue down here at the bottom, it does cover a large volume of brain. And we looked at a mouse that has GFP labeling of the microglia, and that's in green here, and then the red is Evans blue in the vessels. And this is initially the, you can see the surveilling macrophages. This is before sonication. 5 minutes after sonication, however, we see lots of extravasation of the Evans blue, as well as a lot of movement from these microglia. And then if we come back 4 hours later, a lot of the Evans blue is gone, but we see that these microglia are still clamped around the vessels, and they're still helping to respond and reseal the blood-brain barrier. Okay. This is a really interesting video. This is the 5-minute post-sonication time point. Again, what I want to point out here is there is a disparity in the different responses based on vessel size. And what you can see here is that the microglia really have this robust response to smaller vessels. Now, why this is a little unclear. It could be physiological differences between the vessels of different sizes. It could be something simply as physical as the microbubbles having a given diameter in a smaller vessel that represents a larger fraction of a small vessel's lumen than a large vessel, and they may be exerting different mechanical forces on these vessels. So this is an observation that is of interest to us, but it's unclear whether this is physical because of the microbubbles, or there's some physiological difference in the vessels of different sizes. Okay. If we come back 72 hours later, what we see here is that many of the microglia are now orange. So they have endocytosed or phagocytosed the Evans blue, and they are clearing out the blood products that have crossed the blood-brain barrier, and that this goes on for several days and occurs both in the parenchyma down here, as well as we show this as in the meninges occurring as well. Okay. So what causes microglia to respond this way? Well, one of the things and one of the damps that it may be responding to is extracellular ATP. And this is a really nice study from Macon Niedergaard at the University of Rochester, where they used laser ablation with two photon confocal, and they showed that there is this purinergic receptor response where you get colocalization of the GFP with the purinergic receptors, as well as E-cadherin, but not occludin. So what it looks like here is you can see this video where they give a laser ablation, and then right there is where all of the microglia converge in response to the ATP. And if we look at the bigger picture here, ATP is central to a number of the observations that we make. For example, we get a lot of IL-1 signaling to induce inflammatory gene changes. We show that a lot of this happens via NF-kappa B. We activate the inflammasome. We attract basically extravasation of external monocytes or macrophages into the brain, as well as initiating reactive astrocytosis and activating microglia, which then will migrate towards the sites of blood-brain barrier disruption. Okay. One of the other things that we have really been interested in is to compare focused ultrasound and these effects to a more established blood-brain barrier disruption technique. So we were collaborated with Ed Newelt and Leslie Muldoon in Oregon to do hyperosmotic disruption. And what you see here is that they've been doing this clinically since the 80s. And what we did here was to do a unilateral disruption where the entire hemisphere is disrupted. And there is a little bit of contralateral hemisphere effect. This is probably from the mannitol going through the circle of Willis and disrupting a little bit of the contralateral side. But by and large, this is a much more robust disruption than we see with focused ultrasound. And accordingly, if we do a lot of the proteomics and the transcriptomics, we see similar sized effects where here in the ipsilateral hemisphere, we get a proteomic response that lasts more than twice as long as it did with focused ultrasound. And we also interestingly see a contralateral hemisphere response. And I want to point out that while there is visibly disrupted areas of the contralateral hemisphere, all the sampling for these measurements were taken out of areas of the brain that had no visible Evans blue. And so in spite of the fact that there's no clear disruption, those regions of the brain are still experiencing immunological changes. And this could be due to a couple of different things. First of all, we see a large dumping of cytokines and chemokines into the serum following mannitol. This is something I didn't show on the focused ultrasound side, but just to know, we've never been able to detect any analogous changes in serum concentrations of any of these molecules following focused ultrasound. So this contralateral effect could be the effect of these components in the serum, basically activating from the blood side of the blood-brain barrier, as well as there's likely these things are permeating through the G lymphatics, and so you get a lot of diffusion over or movement over to the contralateral side. And so there are potentially two different ways that the contralateral hemisphere is being affected. And we don't see any of this in the focused ultrasound disruptions. We did a lot of transcriptomics here. Again, from an inflammatory standpoint, we do see qualitatively a very similar molecular pattern as focused ultrasound, but it's just that the magnitude of it is substantially greater with the osmotic disruption. And if we look at the histology here, again, we see the same patterns of cellular activation. We get upregulations in IB1 and GFAP. And again, interestingly, this magnitude here becomes important, where out 96 hours, we still have lots and lots of, and what's really, I mean, an increasing activation in both astrocytes and microglia, as well as then cellularly, we see the consequences of contralateral activation at these later time points. Okay. One of the other things that we've looked at is the biological consequences or the long-term effects of blood-brain barrier opening. Okay, and we've done this in a couple of different ways. First of all, we did this what we call Group A. This is we give everybody BRDU, and then we sonicate them. And then in this first group, it's a single sonication, and then we allow them to go out 13 weeks, and we do imaging studies along the way. The other group is something similar to a lot of the clinical trial studies that Suzanne mentioned, where these are repeated disruptions. So we disrupt every week for 1 week, or I'm sorry, for 6 weeks, with imaging studies on week 3 and then week 7. Now, these MR studies were done at 9.4 T. And what we see here, that if a single sonication, we can watch the evolution of these responses. And these actually look okay. I mean, there are a few noticeable abnormalities on the T2 star, as well as the anisotropic imaging does show some changes. But overall, these things are fairly minor. We did do glucose test. This is a spectral imaging technique that allows us to identify certain metabolites based on their unique NMR signatures. And this pretty much we got variable results. We did confirm this with FDG PET studies as well, and they were negative. So from a metabolic standpoint, we're not sure that there's a lot really going on. We are unable to identify anything in particular going on. And one of the things that I think that is important about this study and this slide is that some of these things we can appreciate at 9.4 T, but at 3 T don't appear at all. And so it is interesting and important, I think, in a lot of these readouts and follow-ups is to, there is a place for high field imaging in some of these patient studies. Most of the follow-ups, or at least the vast majority of the follow-ups in clinical studies, are all done at 3T. And we do show that there are things occurring that we just can't appreciate at the lower field strength. Again, here is a histological summary of this. And if we look at this left cortex here, this is the treated cortex, more or less look normal. There are some Prussian blue positive findings in this left cortex. So Prussian blue detects iron, which could be red blood cells or macrophages that have phagocytosed red blood cells, or any iron, any source of iron. But again, like I say, this is this demonstration here that histologically and at 9.4, we can identify some abnormalities that really just are not appreciable at the lower field strength. Okay, if we look at the other group, that this is now multiple blood-brain barrier openings. So this is one blood-brain barrier opening per week for 6 weeks. Microscopically, the histology doesn't look that bad, but we do see enlarged ventricles as well as a lot of dilated vessels in the treated cortex here. And so for this particular group, we treated both this left cortex and then we treated a region over the right hippocampus. So we were able to actually look at both of those changes. And what we see here is that This on the left is the T1 imaging, so for gadolinium extravasation. And what we noticed, and this is simply an artifact of doing a preclinical study in rats, that as the rat ages through the study, the skull thickens, and basically we require more intensity on the ultrasound side to produce disruption. What you can see is that's pretty clear here. We start out at 0.3 megapascals, and the first week we can really get pretty nice disruption, but by the third week we're not producing much disruption. So we had to actually up the power to 0.5 megapascals in order to produce some disruption. And then even then, by the time we get to the 6th week, there's really not a lot of disruption going on. So if we were designing this study to, say, deliver drugs or get a robust blood-brain barrier opening, we are actually undershooting our goals in that regard. Yet, in spite of that, there are now appreciable abnormalities at 3T. So in the 3T, we do see T2 star weighted abnormalities, especially in the hippocampus here, as well as they are certainly visible at the 9.4 scans. Here, if we look at this histologically, what we see is that, and this is now week 7, so this is 6 weekly sonications, followed by 1 week off, and then we harvest the tissue. This is 1 week post the final sonication. And what we see here is that, again, there is some Prussian blue artifacts here that enlarged venules, and then some IB1, as well as some BRDU positive cells, indicating that there's cell division going on. So just to summarize these long-term effects is that, right, with a single one, we see more or less normal-looking outcomes. There is some, there's some variability, but with the multiple BBB openings, we do see substantially more abnormalities. Now, I do want to point out that this, like I say, there are a lot of differences between this study and some of the clinical studies that Suzanne presented on, where they are going to require and necessitate multiple BBB openings. First of all, the clinical instrumentation is infinitely more sophisticated than the preclinical instrumentation is. And so what this really represents is this study that I'm showing here is sort of a first pass of things. Do I think this is the ceiling for what we can do, or this is inevitable for multiple BBB openings? Of course not. I think that there is a lot of optimization that is required, but I think that we have a long way to grow in what we are ultimately capable of doing. Okay. So with all of that being said, we come back and we say, we produce this sterile inflammatory response. And, at first we might think, well, this is a bad thing. And it might be, it might not be. And this is to point out, this is a study, this is by Jürgen Goetz at Queensland, and Suzanne showed many of his follow-up studies, but this was the very first study where he used what's called this scanning ultrasound. It's just a different flavor, but it basically is the same, produces a very similar blood-brain barrier disruption. And what they in fact showed is that this blood-brain barrier opening alone reduced AD plaque burden, and that it required microglial activation. And so from that point of view, these molecular responses are a critical component of the therapeutic response that you're going to see when the blood-brain barrier opening alone contributes to improved outcomes. And as someone asked about tau, there is also preclinical observations that blood-brain barrier opening alone reduces tau burden. So this is Eliza Conofago's group at Columbia showed that the tau projections basically shorten following blood-brain barrier opening. And the mechanism behind this really is not clear, but we have some data. Here on the left here, this is RNA-seq data. Now, granted, this is bulk RNA-seq, so I can't say what particular cells this is happening in, but these changes represent increased expression of genes that stimulate autophagy. And so we see that these persistent changes lasting a couple of days in the brain following focused ultrasound. And what we get here is that if there's neuronal autophagy increasing, which I suspect there is, that becomes a mechanistic explanation for could in part explain why the tau burden decreases following blood-brain barrier opening. That again, it's this sterile inflammatory response is modulating cell biology to yield some therapeutic benefit in the pathology. Okay. The last thing, I don't want to talk too much about oncology in this talk, but this slide does also represent why these sterile inflammatory responses are critical here. And that if we look at tumors, so this is taken from a paper that we published on tumors outside of the CNS, but the analogous strategy would be the same if you wanted to treat CNS tumors, where we use the focused ultrasound to shift the immune landscape from what we would call a cold or immunosuppressive tumor to a hot tumor, which is has an active inflammation. And for this particular study, we just used the focused ultrasound alone. There was no microbubbles, but the approach with blood-brain barrier opening would be the same, where you would use microbubbles to open the blood tumor barrier and then use the extravasation of blood components to initiate an immune response. And whether that would be therapeutic on its own is debatable in a tumor case, but certainly the hope would be that it is an adjuvant that would improve various immunotherapies, whether these be antibodies or CAR T cells. You can have these kinds of things that where we try to prevent immune silencing or immune suppression in the tumor itself. Okay, so I think that the take-home message to a lot of this is that there is going to be an enormous amount of optimization that is required to make focused ultrasound clinically useful. Okay, these are a couple of nice studies done by Mark Borden at CU Boulder, where they're looking at biological readouts given various alterations in conditions. And I won't go through all of these, but basically we can boil this down to where it is the magnitude of the blood-brain barrier opening, which dictates the molecular and cellular responses, and by extension, drug delivery. If you're just looking to deliver a particle or a drug, and I don't even care about the molecular response, I'm still going to be concerned with the magnitude of the blood-brain barrier opening. And this is really dependent on many parameters of the ultrasound, because remember, this is an ultrasound converging with a microbubble to generate an effect on the blood vessels. And things like, say, frequency, intensity, how you deliver the pulses may be important, as well as the bubbles here. So, chemical composition becomes a big deal, size, the dispersity of the population. And, one of the things is there is an enormous amount of research being done right now in optimizing bubble formulations and bubble populations. That being said, from a clinical standpoint, I think there are just some regulatory and really economic hurdles right now that I'm not sure how quickly any of those are going to be in the clinic. And thus, we have to make do with the 3 FDA-approved bubbles that were designed as as diagnostic contrast agents, and they weren't really designed for blood-brain barrier opening. Okay, but how these things coalesce to produce this is really being widely investigated right now and is really urgent. I will throw in one additional thing that I think will create a lot of variability for blood-brain barrier opening, and that is going to be the physiology and the pathology of your patient. One of the things that we started in, and this is, like I say, again, a very budding nascent line of research in our lab, but I'm interested to follow it and see where it goes because I think it may portend very important things. Is that right here on the left? This is again the graph of cavitation here, and just to remind everybody that cavitation is this compression and expansion of the bubble so that it pushes against the vessel wall. Now, if you increase acoustic pressure, you do get bubble collapse and is essentially an explosion, which is destruction. And this, we want to avoid. This is sort of almost universally recognized as destructive to tissues. But this can be controlled, and we can limit the cavitation activity to, as Suzanne mentioned, is what's called stable cavitation. Okay, but what is it doing to the vessel walls? What is it doing to the endothelial cells? And I think there is an urge to assume this is simply a physical disruption, that the expansion of the bubbles pulls tight junctions apart. But interestingly, if we just go to a very simple transwell system, so this is, we can culture microvascular endothelial monolayers in these transwells, and we measure transendothelial electrical resistance, where high resistance indicates that tight junctions are formed and there is not passage of ions across the monolayer. As the tight junctions are disrupted, the resistance drops as you pick up current. And what we see here is that we did just a really simple experiment where we start and we measure the tear during sonication, and we see that there is, while we're sonicating with microbubbles, an expected drop in resistance. However, we shut off sonication, and there is now no longer an interaction between microbubble forces and the cells. We see this continued statistically significant decrease in electrical resistance, suggesting that this region of the graph is indicating something physiological going on that is remodeling these barriers here. Okay, we have done a whole host of work on how calcium dynamics work during focused ultrasound. This is here sort of a general schematic from our most recent paper that incorporates about everything we know at this point. And one of the things that we discovered is that in multiple cell types, and as Suzanne mentioned, voltage-gated calcium channels are critical to maintaining this calcium signaling. Now, there are some theoretical descriptions of ultrasound directly activating voltage-gated channels, but there's not a lot of experimental evidence. What we demonstrated was, in fact, that there are in the plasma membrane populations of these voltage-gated calcium channels that are complexed with mechanically sensitive channels. And so the focused ultrasound, or the microbubble, pushes on that, and that activates a current that causes just enough depolarization in the local area to open the voltage-gated calcium channel, which then which opens the voltage-gated calcium channel, and then it allows enough calcium in to propagate calcium-induced calcium release. There is also purinergic receptor activity that stimulates the formation of IP3 and additional calcium release. Okay, so when we think back to endothelial cells here, what we know is that calcium is critical for remodeling of a lot of these tight junctions. And so here what we do is, this is, again, these same microvascular endothelial cell monolayers, and then we labeled it with Flow4 as a calcium indicator, and then are able to do calcium imaging with them. And you see during sonication, we get a pretty robust calcium response. And I do want to point out that we did do cavitation monitoring during this time, and we don't see any spectral evidence that destructive cavitation or inertial cavitation is occurring. So this, we feel, models pretty much what you would see in the inside of a vessel, where we stimulate a lot of calcium transients. Okay. If we look at how tight junctions behave following this kind of treatment, what we see here is that in the control condition, we get a lot of tight junctions, ZO1, which is also called tight junction protein 1, and that if you give focused ultrasound, 30 minutes later, we get a reduction in the expression. And this is consistent with really what was known at the beginning of the talk, before we even got involved in this, and that it is transient. They do come back after a couple of hours. Now, interestingly, we can block this effect by chelating calcium with EDTA. So we chelate extracellular calcium. So you don't allow any calcium in, and it blocks this rearrangement and this remodeling of tight junctions. As well, we can use verapamil. So this is an L-type calcium channel blocker, and we see that, again, the effects of ultrasound and microbubbles are really attenuated when you pharmacologically inhibit processes in the endothelial cells. We can also look at how actin remodeling works. And we see here that, again, in these monolayers, this is an unsonicated. You get these nice networks of actin. Depolymerization occurs, or well, it's depolymerization and repolymerization probably. But again, remodeling, I guess, would be the right term for it. And again, that these are blocked by intracellular chelation of calcium using BAPTA, as well as blocking channels using amlodipine. So a different L-type calcium channel blocker inhibits these rearrangement of actin. And if we look at the effects on endothelial resistance in the layer, we see that the decreases in endothelial resistance, which reflect opening of the tight junctions, is inhibited when calcium can't enter the cell, and specifically when the calcium cannot enter the cell through the L-type calcium channel. So this brings up a slightly different perspective of things. And like I say, again, this is a very novel line of research in our lab, but I'm really, really excited to see how this affects things. That if we look for our molecular responses or our drug delivery magnitude, how do pathologies that affect endothelial cell calcium signaling pathways affect blood-brain barrier opening? So this could be, for example, in someone who has atherosclerosis. Also, one thing that I'm really interested in is, again, this blocking L-type calcium channels inhibits the cellular processes that would be required for blood-brain barrier opening. And so what if you have hypertensive patients, and we'll say, to make things easy, their hypertension is well controlled, but you have a patient who has it controlled with a channel blocker versus someone who has an ACE inhibitor or a beta blocker or something like that, that wouldn't affect endothelial cells in the same way. Would those different groups of patients respond differently to the same ultrasound and microbubble conditions? Would we still produce variable opening, variable molecular and cellular responses, and variable drug delivery? So I will leave everyone with that question. I will be back in the lab trying to answer that soon. If anybody'd like to help, we are more than happy to collaborate with anybody. So with that, there are several acknowledgments that I need to make. Obviously, Joe Frank, as the chief of the lab, has been fantastic to work with. That's why, as Christy said, I never left NIH, and he's a big reason why. Really, really enjoy working with him. A number of really, really talented fellows, both past and present, that have come through the lab, and our collaborators, Dima Hamoud, who does PET with us, Dorian McGavin, who did the 2-photon confocal microscopy, and then Ed Newell and Leslie Muldoon, who helped us with the osmotic disruption. So with that, I thank everybody for their attention. I'm happy to take any questions you might have.
I really enjoyed your presentation. Beautiful work.
Thank you.
So, putting the two talks together, now I'm wondering as to what the mechanism of clearance of the amyloid might be, and if that microglial cell activation may actually be playing a part. What do you think?
Absolutely. I think that right, without microglial activation, we just don't get much clearance of plaques. It's really only following extravasation of the blood components that stimulate that activated phenotype before we really see any actual activation, or any endocytosis of the plaques or phagocytosis of the plaques.
Okay, the second question I had is that one of the challenges in treatment of various neurological diseases is the delivery of antisense molecules to the brain. And if anybody's ever used focused ultrasound in order to deliver them?
I'm not aware of anybody using antisense directly, but there is a big initiative to get viral vectors across, and maybe viral vectors express shRNA or something like that. So certainly the delivery platforms people are working on, but I'm not sure exactly if people are getting antisense oligonucleotides specifically across.
All right, thanks.
Hi. You're obviously from Joe's group, and Jennifer was in our group in neuroimmunology. I've been working with Alfonso's world.
Pardon?
Alfonso Silva in Pittsburgh, and David Schaefer, who's doing a lot of focused ultrasound in marmosets. And we've got a great quality of marmoset. I mean,
I know. Yes,
and it's kind of the extension of this study in the rodent model to something we could do actually here at the NIH, with the imaging systems we have. And I was wondering with you or Joe he's gone, but it's an opportunity.
That would be fantastic. And, one of the things is that I have sort of been advocating over the years that, we really need, if we want to see focused ultrasound here at NIH really take off, is that we do need to start gathering all of the relevant expertise, and like I say, I remember, seeing, a lot of Jenny's images from the marmosets. I mean, absolutely beautiful stuff. And yes, we would love to start branching out and being able to assess not only imaging and molecular biology, but behavior, and really start to comprehensively look at these things. And I'll make a point on neuromodulation, for example. There are some very impressive results, but it's totally unclear really how we're doing it. And, I think that there's a big opportunity for basic and translational science to step in and really fill some of those gaps. And I think, like I say,
because the Pittsburgh group, David, they're pretty much focusing on Alzheimer's, right?
As far as I know, yeah.
And I've seen that data, and we come in our group in more of a neurological, and exactly kind of how you set this up. So it seems fairly, we'd be interested very much, us and Dorian and all, you've got a really good group of neuroimmunologists here to look at, the ability of immune cells to enter T cells, Tregs, antigen specific cells, B cells. There's a lot of work we're doing, obviously, on the immunological side, but to kind of couple it, with this kind of neuromodulation, as Avi was saying, how to deliver these, sometimes we think they're bad guys, but sometimes we think they're pretty good guys.
Sure.
That we need to get up there. This would be a really great opportunity. I'm just offering that. I mean,
it would be fantastic. We are always
something to think about.
I am always interested in collaborating. Like I say, if anybody has ideas and expertise, like I say, our problem is we have too many ideas and not enough hands, not enough time. But yes, like I say, we are really investigating, and I with like Brad and Joe, Brad Wood, talked a lot about really centralizing the expertise here at NIH that would be necessary to make a meaningful program happen here.
So ultrasound can be used in a variety of different intensities. You can do a tiny bit and modulate the physiology. You can do a little more and get a transient opening. You can blast it, and you can get cavitation. Yeah. Now you're sort of in the middle there. The question is if you really blast it and get sort of cavitation, how does that link with what you're doing? Do you actually get a big microglia response if you blast it? I mean, I would anticipate so.
Absolutely. However, it's a condition we most often would like to avoid. Certainly that is associated with, extensive tissue damage. And, even if you want to do tumors, for example, destroying the vessels may not be a good idea, right? You may squander your opportunity to continue delivering other therapeutics, especially from the point of view of the immunological changes in tumors. We don't see focused ultrasound being a monotherapy in that regard. It would be an adjuvant to other things. And so even in that context, outright tissue destruction may or may not be something you'd want. Outside of that, we are not looking to destroy tissue. Yep.
Thank you very much. It was a fantastic talk. I have a question about lung. Have you looked at the neuronal apoptosis or neuronal damage in your model, especially in the repeated model and the long-term model? Because we know that there are a lot of literature on the pro-inflammatory role of microglia in the neurodegeneration and neuronal death now in the Alzheimer or neurodegenerative diseases. Now we induce some kind of burst of neuroinflammation, and you look at the kind of a role in the microglia. How about the neurons? Have you looked at the DNA damage, apoptosis?
Not specifically. I mean, sort of, you look at like the tunnel staining issue, but I mean, that's all CNS cells that, we're not focusing on neurons specifically, but yeah, I mean, it's an important aspect. And I think, again, it's, the molecular neuroscience tools are probably a little more sophisticated than I'm ready to jump in and use right now. But yeah, it would be great to start looking at those questions and understanding those impacts.
Yeah, thank you.
All right. Well, thank you very much. And like I say, I really enjoyed being here. Thank you very much.